One of the activities we perform is testing food and feed products for the presence of genetically modified organisms, or GMOs. In the European Union, some of the GMOs are approved, but others are not authorized for the European market. So how do we know if food or feed materials contain GMO-derived materials, and if they are approved? We begin testing as soon as the food samples are delivered to the lab. These samples are carefully registered so that the entire process can be closely monitored. The sample is then ground in order to homogenize it. Every single bit of it should give the same result. Next, we isolate the DNA from the homogenized sample. We now have what we need, a strand of DNA that can tell us whether or not it contains GMO material. If a crop is genetically modified, then a sequence of genes has been added to the DNA. This sequence leaves a trace, and it's that trace that we look for. In order to find this trace, the pipetting robot prepares the next step, the detection and identification of the GMO-derived sequences. Next, the polymerase chain reaction, or PCR, takes place. Let's take a closer look at what's really happening. The double helix of the DNA is first separated into two strands. Then we need two primers, a starter primer and a stop primer. The primers attach themselves to each strand of DNA. Afterwards, a special enzyme starts completing the target DNA, using the free nucleotides that we've added to the mixture. By repeating this process, we can produce more than 1 million pieces of DNA in just a few hours. We can now make the DNA visible and quantify it with fluorescent chemicals. All the GMO-related sequences that we identify this way, we compare with information on GMOs that we have in our databases. In most cases, this leads to a positive identification. However, sometimes we find GMO-related sequences that we cannot explain, and that may indicate the presence of unauthorized GMO. In that case, we perform another PCR reaction. Now we start from the identified GMO element into the unknown adjacent sequences. This way, we achieve an extension of hundreds to thousands of bases. That may be sufficient to identify an unknown, unauthorized GMO and provide some information about the hazards it may entail. Today, we perform this process for one GMO element at the time. However, with the help of national and European partners, we are developing new technologies that enable us to perform multiple analysis. Any trace of GMO we find, we report it back to our client so that appropriate measures can be taken. That's how RE contributes to GMO transparency in human and animal foods.